Hi, thank you for coming today. So I'll be talking today about my project that I did this summer, which was the characterization of tomato sugar transporter SWEET10. I'm actually going to dim the lights now. All right. So first, I'm going to go over some background on sources, sinks, and sugars. A source is a photosynthetic tissue, the leaf, that produces photoassimilates, such as sugar, which are transported and stored in sink tissues, such as the roots, the shoots, and the meristems, and specifically in our focus today, the fruit. Sugar plays an integral role in the signaling, growth, and development of fruit, especially in early stages. And we are interested in studying the early development of fruit and are using tomato as our model system. So here I have a profile of the sugar accumulation in tomatoes throughout development. We have our tomatoes developing up along here. And sucrose is the main photoassimilate. It is transported from leaves to the fruit, which I'll actually talk about more on the next slide. But throughout tomato development, it is broken down into glucose and fructose. Glucose is then also stored as starch, which accumulates throughout development, before just the onset of fruit ripening, where it is broken back down into glucose. So back to our sucrose transport from the leaf, our source, to our sink, the fruit. We have our sucrose molecules being transported into the sieve element of the phloem, where it is eventually transported and unloaded into the sink. This unloading can occur in one of two ways. We have symplastic unloading, in which sucrose is transported into this area, called the symplast, through plasmodesmata. Or we have apoplastic unloading, in which the sucrose is transported into the apoplasm, this area of the cell, using a transporter, such as soot or souk. A recent family of transporters, the sweet transporters, has been characterized in the plasma membrane of the leaf parenchyma cells. The sucrose is transported into the sieve element and is the first characterized step in phloem loading. Today we are interested in seeing if sweet plays a role in the actual process of unloading into the fruit. Now, I just want to real quick say that sweets don't just transport sucrose. They transport other sugars like glucose and other hexose sugars. And the sweet gene family is broken into 4 clades. Today we are talking about clade 3 because it is known to transport sucrose. A recent study in our lab using RNA sequencing identified SWEET10, a gene that is highly expressed in fruit during early developmental stages. We can see here that the internal tissues, the placenta and columella, are especially high at enthesis and 5-DPA. Enthesis is the opening of the flower. DPA is days post-enthesis. So with this high expression, we can see that the placenta and the columella, circled here, they are very high. They're very rich in vascular tissue. Altogether, this makes SWEET10 a very promising candidate in playing a role in the unloading of sugar from the phloem to the fruit. So I hypothesized that tomato SWEET10 is involved in sucrose transport during early development of fruit, and that SWEET10 is localized in the vasculature of the tomato fruit here. This summer, my aim was to functionally characterize the SWEET10 in tomato fruit using 2 different approaches. The first approach being a characterization of the expression of SWEET10. I used transgenic lines, 3 different types, which are the 35S constitutive promoter used to overexpress SWEET10, RNAi used to knock down expression of SWEET10, and CRISPR used to knock out expression. My second approach to characterizing this this protein was through the identification of the subcellular localization of SWEET10 in the fruit, using a translational fusion with YFP. So just a brief overview of my characterization of the gene expression. The transgenic lines for 35S, RNAi, and CRISPR had already been generated when I had gotten here. I monitored fruit growth and collected placenta and columella tissue to be used to quantify gene expression using reverse transcription quantitative PCR. And this tissue is also used to quantify the sugar accumulation in these tissues. So I was looking specifically at starch and soluble sugars such as sucrose, glucose, and fructose. These sugars were quantified using an enzymatic assay. So first, just the molecular characterization of my transgenic lines show that our overexpressed lines, the 35S, have a higher relative gene expression compared to the control, which is good, that's expected. And then our RNAi lines, which are, again, the lines that have knocked down expression of SWEET10, show, again, a lower expression of SWEET10 compared to the control. Our CRISPR lines that have the knockout of function, the mutations causing this knockout are a deletion in the promoter region and exon 1, that was a 198 base pairs deletion, and then a 1 base pair deletion that caused a frame shift and a premature stop. So after I characterized that this SWEET10 is actually different in expression, I was able to move on to observations on the phenotype from these transgenic lines. And interestingly, while we were growing it, the first thing that we noticed about SWEET10 transgenic lines was that it appears to affect tomato fruit growth. We can see that 35S lines, our overexpressed lines, have a lower fruit weight and are visibly smaller. The opposite can be said about CRISPR and RNAi, in which we have a higher fruit weight compared to the control, and the fruit is visibly larger as well. This is the first proof that SWEET10 has some kind of effect on fruit development. Next, I characterized the actual function of SWEET10 in transporting sugar in fruit, and I collected tissues at 5 dpa, 10 dpa, and 15 dpa, and compared the percent difference from control. With our 35S lines, we have a lower accumulation of glucose, and with our CRISPR lines, we have a higher accumulation of glucose. This trend can also be seen with sucrose, fructose, and the opposite trend is seen with starch, in which our CRISPR lines show a lower accumulation. Now, I also tested the sugar accumulation for the RNAi lines, but they showed basically the same results as CRISPR, so to save time and space, I'm only showing you the CRISPR accumulations. This actually supports our first hypothesis that SWEET10 expression does affect the transport of sugar in fruit. So next, I then characterized the localization of SWEET10. We were looking at where in the cell this protein is actually functioning. Previously, a transgenic plant had been generated with a YFP fusion to the C-terminal end of our SWEET10 protein. I used confocal microscopy and stereo microscopy to image the fruit from these plants. Interestingly, well, not interestingly, this is actually along the lines of all of our experiment, we see that fluorescence is strongest in the early stages of development of these fruit. Here I have a large slice of the tomato fruit that I want to direct your attention to the vasculature, because this is where we are expecting SWEET10 expression to be. This is exactly what we saw when we looked at our slices. We have the vascular complex, these lighter areas. They're not holes, they are tissue. And we see that our YFP tagged SWEET10 is actually localized in the vascular complex. But interestingly, it's not in all of the cells of the vascular complex. It is in very specific locations. So here we're looking at the equatorial slice of the tomato, so we're looking like this. And then these slides, we're looking at a longitudinal slice, so we're looking like this. You can see here, it's a little harder to see the vascular tissue here, and up close, the vascular tissue here, this possibly being a sieve element for a phloem. And then we see that SWEET10 is, again, localized around the vascular tissue and in very specific cells. So these results are very exciting because the localization of sweet proteins in fruit has never before been identified. This novel finding will help us in understanding the overall role that sweets play in sugar transport, and specifically SWEET10. So in summary, this summer I have identified an inverse relationship between SWEET10 expression levels and the fruit weight, as well as the accumulation of fructose, glucose, and sucrose. We also have a positive relationship between SWEET10 expression levels and starch accumulation. And finally, SWEET10 is located in the vascular complex, appearing to be specific to certain cell types. All right, so I've made a model for the function of SWEET10 in the process of phloem unloading. We have SWEET10 here on the plasma membrane of the phloem parenchyma. And when it is functioning properly, we have sucrose being transported into the symplast, where it is eventually broken down and can be stored as starch. However, when SWEET10 function is knocked down or knocked out, we see that there is an accumulation of sucrose in the apoplast, where apoplastic invertases will break it down into glucose and fructose here. And this explains why we're seeing a higher accumulation of these sugars in our CRISPR and RNAi lines. Because there is a lack of sucrose being fed into the symplast, we do see a decrease in starch accumulation, again reflected in our RNAi and CRISPR lines. So this model is not complete. We still have some things that we want to study, such as the very specific cell in the vascular complex that SWEET10 is located in. So we want to try to identify what part of the vascular system it is in. And my lab also aims to do a sugar transport assay that will test the specificity of SWEET10 sugar for other sugars besides sucrose. So this is whether or not it will be glucose or other hexose sugars. I would really like to thank Carmen and Philippe for being wonderful mentors. This summer has been great. I'd like to thank Delaney and George for making this program possible. I'd like to thank the BTI facilities, specifically the Plant Imaging Center and the greenhouses. And I would like to acknowledge the funding sources that have allowed me to be here this summer. I now have time for any questions. Thank you for listening. Okay.
When you describe the GLP construct, the transgene, what was the promoter that was used for that transformation?
No, for this transformation, it was the natural promoter. We didn't want to do an overexpression because then it would be expressed in all tissues of the fruit, and we wouldn't really be able to see where it was naturally.
And my second question is, do you have a hypothesis regarding the size of the fruit that was like bigger in the CRISPR lines and smaller when you overexpressed the gene? Like why the size was so different?
So there are a lot of things that can be affecting this, but one of the hypotheses there is that a higher level of sugar in the cell or in the fruit can cause a difference in osmotic pressure, which can affect the size and the growth of the cells. Okay, I saw. All right, Josh, go ahead.
So does not sweet actually make the fruit sweeter?
That's hard to say because what we're seeing here, let me go back to my model. So this is the entire, knocking out sweet will just cause an accumulation of these sucroses in the vascular tissue. And so I don't know how it would make the entire fruit taste, but it's more about the transport of the sugars from the vascular tissue of the fruit to the other tissues of the fruit.
It looks like the highest expression level for SWEET10 was in flour. Did you if there was any effects on flour morphology or any effects on the flour?
Overall, no. Generally, the RNAi lines, I believe, took a little bit longer for everything to develop, but there was no real visible change.
So there were two methods of gene silencing that was used. Was there a preferred method that you found, or methods?
Well, we used the RNAi to knock down the expression, so there was still just low levels of expression, and CRISPR was used to completely knock out expression. So we used that just to be sure that it was kind of an incremental change. And from what I could see, there were no real effects or differences in between RNAi and CRISPR.
Okay, I think we're out of time.
All right,
Thank you.
Thank you.